Thanks very much. I'd like to thank the organisers for inviting me to this absolutely fantastic city and to speak at this very significant meeting. The Pancreatic Research Group, of which I'm a member in Sydney, has actually been in existence since 1965, so we're 51 years old. Originally, we concentrated on the pathogenesis of alcoholic pancreatitis. In 1998, as has already been said, we were lucky enough to be the first people in the world to isolate pancreatic stellate cells. And subsequently, for the last 17 years, we've been looking at the pathobiology of stellate cells, particularly as it relates to alcoholic pancreatitis, but also in relationship to pancreatic cancer. Alcoholic pancreatitis is a major cause of acute pancreatitis in the Western world and the predominant cause of chronic pancreatitis in the Western world. When you're dealing with alcoholic pancreatitis, you're dealing with a clinical paradox. On the one hand, the risk of developing pancreatitis increases with increasing doses of alcohol. The more you drink, the more likely you are to get pancreatitis. But even those very heavy drinkers, not all of them develop pancreatitis, less than 5%. So there is a susceptibility or trigger factor in this disease as well. It goes without saying that you need alcohol to define alcoholic pancreatitis. If you don't drink at all, you don't have alcoholic pancreatitis. How much do you need to drink to get alcoholic pancreatitis? A lot. The isolated alcoholic debauch rarely, if ever, causes pancreatitis. Heavy chronic consumption of alcohol is required. Early epidemiological studies, this one from France, suggested that the relationship between heavy consumption of alcohol, the amount of heavy consumption of alcohol, and the risk of pancreatitis was linear, from 0 right up until very high levels of alcohol consumption. However, recent epidemiological studies have suggested there is a threshold, and the risk is negligible in those who drink less than about 5 standard drinks a day and increases substantially thereafter. Given that there is a clinical paradox to this disease, we've adopted 2 approaches in the past to study the pathogenesis of the disease. The first approach has been to study the constant metabolic effects of alcohol or ethanol on the pancreas. And I guess copying the way in which the liver doctors try to elucidate the pathogenesis of alcoholic liver disease, we looked initially at ethanol metabolism. Now, ethanol metabolism in the body can be via 2 pathways, the oxidative pathway or the non-oxidative pathway. The oxidative pathway involves alcohol dehydrogenase, aldehyde dehydrogenase, cytochrome P452E1. It results in acetaldehyde, which is toxic, and it results in the production of reactive oxygen species, which cause oxidant stress. The non-oxidative pathway involves the esterification of ethanol with fatty acids to form fatty acid ethyl esters, and it's thought that this pathway may be more important in the pancreas than in any other organ with respect to tissue damage. We studied pancreatic ethanol metabolism in isolated Acinar cells, studied the group from Steve Pandol's group from Los Angeles, and we found that the major pathway for ethanol metabolism by pancreatic Acinar cells is the oxidative pathway, with comparatively very little in the way of fatty acid ethyl esters being produced. Nonetheless, the fatty acid ethyl esters that were produced in sufficient amounts that our in vitro experiments established could be toxic to pancreatic organelles. Now, there's variation in the way in which alcohol is metabolized depending on the species. Rat Acinar cells express a class 3 ADH, which is a very high Km pathway, whereas human Acinar cells express an ADH class 1, low Km, higher activity. Pancreatic stellate cells, which you've heard today, and you'll hear a little bit more about in this talk, express an ADH class 1 enzyme. With respect to the pathogenesis of alcoholic pancreatitis, there are three types of cells or cellular elements that I want to talk about this afternoon: Acinar cells, pancreatic stellate cells, and duct cells. Now, this is a cartoon of a pancreatic acinar cell showing ethanol down the bottom, causing a number of metabolic changes within the cell. Ethanol increases the content of the cellular content of digestive and lysosomal enzymes. Lysosomal enzymes are capable of activating digestive enzymes, and hence they're capable of initiating autodigestion. In addition, chronic ethanol feeding increases the fragility of zymoid and granule membranes and of lysosomal membranes, thereby facilitating contact within the cytosol of lysosomal and digestive enzymes, predisposing to autodigestion. Other changes that have been described in the acinar cells in response to ethanol include oxidant stress, cytokine release, a sustained increase in calcium, which results in mitochondrial depolarization and death. All these changes together are thought to combine to cause cellular necrosis and a subsequent necroinflammatory response with cytokine release. Of late, there have been interest in the homeostatic mechanisms, the unfolded protein response and autophagy within the cell, and the effect of ethanol on these. And I'll deal with these just in a little while. Now, just next door to the pancreatic acinar cell, there's the pancreatic stellate cell. The stellate cell can be activated by cytokines released by inflammatory cells, and they can also be activated by directly by ethanol, which causes oxidant stress via its metabolism by the cell. So there are two cellular elements. The third cellular element is the duct cell. This has remained sort of relatively unattended, and not many people paid attention to it until Peter Hege's work in Hungary a few years ago, published in Gastroenterology. And the Hege group showed that ethanol decreases CFTR activity of ductular cells and decreases CFTR expression, thereby possibly sort of interfering with pancreatic secretion and making it more concentrated and more viscous. Now, I alluded to two homeostatic mechanisms that have recently received attention. The first is ER stress, or the unfolded protein response system. This is a complicated diagram. I use it just to emphasise that cells that produce a lot of protein are particularly on guard to make sure that the protein is actually produced in a way and secreted in a way that avoids havoc. And one of the ways that havoc can be sort of wrought upon a cell is when the synthesised proteins become unfolded. And when they become unfolded, they activate a number of sensory mechanisms within the endoplasmic reticular membrane to see if they can rectify the situation. And how do they rectify the situation? Well, they have enzymes that can go in and refold the protein. They have chaperone enzymes that can go in and sort of escort the proteins elsewhere. They have signals that can actually attenuate translation so that the protein synthesis of the cell decreases, turns down. And finally, if it gets out of control, they can activate a death pathway and the cell undergoes apoptosis. Now, this slide is taken from Steve Pandol's work in Los Angeles. They looked at rats fed controlled diets and ethanol diets, and they found that morphologically, the animals fed ethanol diets exhibited more dilated endoplasmic reticulum, as seen there in the right panel. And in conjunction with this change, there was an increase in sliced XBP1 protein, which is actually a protein involved in the unfolded protein response, the stress response. So this is evidence that ethanol by itself can, in acinar cells, promote endoplasmic reticular stress. How about autophagy, another cellular homeostatic mechanism? Autophagy is the mechanism whereby cells cleanse themselves of effete proteins and macromolecules. There is a membrane, a double membrane called a phagophore, which wraps itself around effete organelles such as mitochondria and macromolecules. It then closes with the aid of a molecule called LC32 and matures into what's known as an autophagosome. The autophagosome then aligns itself and plugs into a lysosome, and this requires the presence in the lysosomal membrane of a protein called LAMP2, and as a result you get an autolysosome which gobbles up all these organelles that are destined for recycling. Now, Franco Fortunato and David Whitcomb from Pittsburgh in 2009 provided very good evidence that chronic alcohol intake increases the presence of autophagosomes in acinar cells and also autolysosomes. So there was increased flux along the autophagic pathway. When they added lipopolysaccharide, which is a pancreatic toxin, in combination with alcohol, they got increased autophagosomes again, but a decrease in autolysosomes, suggesting that LPS or endotoxin somehow sort of blocks this homeostatic mechanism. And I'll be talking about LPS a little bit later because we believe that it's a physiologically, clinically relevant trigger factor for human alcoholic pancreatitis. Now, this is a schema that I'm going to build on about the pathogenesis of alcoholic pancreatitis. We have ethanol there on the left, generating its toxic metabolites, acetaldehyde, fatty acid ethyl esters, also causing oxidant stress. These two elements together cause a number of changes within acinar cells: autodigestion, cytokine production, mitochondrial injury, ER stress, and impaired autophagy. And then, under the appropriate trigger factor, these changes result in pancreatic necroinflammation. Now, what is the susceptibility or trigger factor that is responsible for this? At the present, we don't know, but when looking at potential trigger factors, it's important to compare alcoholics with pancreatitis to those without pancreatitis, so that the index and the control groups differ in only one variable, the presence or absence of pancreatitis. Many studies looking for putative susceptibility factors use the general population, which is as a control, which is a less appropriate control than alcoholics without clinical pancreatitis. With respect to susceptibility factors, environmental and metabolic factors that have been examined to date, you can see there are lots of them. Most of the studies have been negative. I want to talk a bit about smoking, which is highlighted there in cyan. A number of studies have been done looking at smoking and alcoholic pancreatitis, and it's now going into the textbooks that smoking is a precipitating factor for alcoholic pancreatitis. But the only studies that have compared alcoholics with pancreatitis to alcoholics without pancreatitis has been the study by Haber and colleagues in cyan and the Yadav study in 2009, second from the end. And both those studies were negative. So the problem with studying smoking and alcoholic pancreatitis is that most studies are retrospective. I think they have to be. And people who drink heavily also smoke heavily, so it's difficult to tease apart the two variables. So, you're comparing it, it's very hard to actually ascertain whether the changes you're seeing are due to alcohol or to smoking. We believe that smoking is unlikely to be an initiating factor for alcoholic pancreatitis, but it may play a role in disease progression. So Mazinerv and colleagues, that's Al Lowenfeld's study in gut in 2005, I think provided convincing evidence that in those people who drank heavily and smoked, they got earlier diabetes and earlier pancreatic calcification. And there have been other studies from Japan and elsewhere that have actually sort of reinforced this notion that smoking tends to accelerate the progression of the disease. With respect to individuality susceptibility factors, putative factors that are inherited, a number of things have been looked at, a number of genes, a number of mutations. Most of these have revealed negative results or results only in a very tiny subset of patients with alcoholic pancreatitis. A particular interest is Claudin-2, which is a gene coding for a tight junction protein. Now, Dave Whitcomb and his colleagues from Pittsburgh conducted a GWAS, a genome-wide association study and published in 2012 in Nature Genetics, indicating that there were Claudin-2 variants described in patients with alcoholic pancreatitis. They did have an alcoholic group without pancreatitis to which they could compare. They believed that the expression of Claudin-2 was increased in acinar and ductular cells. However, Claudin-2 is a tight junction protein in the intestine, and increased expression in the intestine has been associated with increased permeability, although this has not been studied in patients with alcoholic pancreatitis. In the same vein, alcohol can cause in vitro increased permeability of intestinal Keiko cells that are on a dish via its metabolism by cytochrome P452E1, the formation of reactive oxygen species, and the upregulation of the circadian proteins CLOCK and PER2, giving increased permeability. We can only guess or surmise where Claudin-2 might actually fit in this sequence. Now, if there is increased permeability in pancreatitis as induced by alcohol, why is that important? Well, we believe that endotoxin is a trigger factor for alcoholic pancreatitis. Gut permeability is increased in alcoholics. Increased gut permeability permits translocation of gram-negative bacteria, allowing endotoxins to enter the circulation. Plasma LPS or endotoxin levels are higher in drinkers, and LPS is known to be injurious to liver and lungs. So when we feed animals alcohol for periods up to 8 weeks and then at the end of the feeding period give them endotoxin, we find in the pancreas they get necroinflammation. This is a histology score here for the animals given alcohol and lipopolysaccharide or endotoxin have inflammation. They also have fibrosis. Alcohol plus endotoxin gives an increased amount of fibrous tissue in the bottom panel as measured by, as estimated by Masson's trichrome staining. And this increase in fibrosis is thought to be, we think it's due to pancreatic stellate cells because they can be seen here stained in the bottom right panel for alpha smooth muscle actin. Pancreatic stellate cells can respond to endotoxin. They possess the receptor, TLR4, both human and rat pancreatic stellate cells do this, and they can also, in rats' pancreatic stellate cells, they also express the co-effector molecules for TLR4, CD14, MD2. This work done by Shimasugawa's group in Japan. Now, how does alcohol and LPS synergize? Alcohol itself increases intestinal permeability, increases circulating LPS. These are clinical facts. Increased circulating LPS causes TLR4 activation, downstream signalling activation, and the generation of pro-inflammatory mediators. Alcohol, in addition, causes oxidant stress in cells, and oxidant stress can activate TLR4. Alcohol itself can cause premature activation, release of active pancreatic enzymes, including elastase, which has been shown also to increase TLR4 activation. So we're back to this schema of the pathogenesis of alcoholic pancreatitis, and we've got all these alcohol causing all these changes, causing pancreatic inflammation. We believe that endotoxin's a plausible trigger factor for this, and pancreatic necroinflammation is established to be a cause of stellate cell activation via local cytokine release. In addition, oxidant stress produced by ethanol metabolism can also directly activate stellate cells. These changes lead to pancreatic fibrosis. Smoking probably fits into the schema by exacerbating pancreatic necroinflammation and by activating stellate cells. So we have a system in our lab whereby we can feed animals alcohol and put them in a smoke box, a smoke cage, expose them to smoke for 6 hours a day. And when you expose animals to alcohol, endotoxin and smoking, the changes in the histology score in the pancreas are greater, as indicated by the green bars, and they get worse fibrosis. Smoking induces ER stress in these animals fed alcohol and given LPS. There are four pictures here on this slide. The top left-hand one, normal pancreas, alcohol plus lipopolysaccharide, rough endoplasmic reticular, mild dilatation. But look at what happens when you add smoking. The pancreas looks like it's been machine gunned. And those vesicles you can see are actually grossly dilated endoplasmic reticular lumina. In addition, we've shown that pancreatic stellate cells express nicotinic acetylcholine receptors. In other words, they can respond to nicotine in cigarette smoke as well as other compounds in cigarette smoke, including NNK, which is a nitrosamine ketone, and it's an important component of cigarette smoke in terms of its toxicity. So on the left panel, we have evidence that there is expression of the nicotinic acetylcholine receptors, and when you actually expose stellate cells to ethanol and cigarette smoke extract, which is something you can buy from a commercial manufacturer, when you add those two together, there's an increased expression of nicotinic acetylcholine receptors in the stellate cells. And alcohol and smoking together, this slide, look at just at the far right bar, alcohol and smoking, or alcohol and cigarette smoke extract, or NNK, increased the migration or the activity of stellate cells in comparison with cigarette smoke compounds or NNK alone, or alcohol alone. So in summary, in the modern era, our concepts about alcoholic pancreatitis are complex. Alcohol exerts constant metabolic effects. I hope I've shown this on acinar cells, duct cells, and stellate cells. A trigger factor is required to produce overt disease. Endotoxin is a likely, clinically relevant trigger factor. Susceptibility may be mediated by factors causing increased intestinal permeability, such as Claudin-2 and the circadian proteins CLOCK and PER2. Smoking adds to the toxicity of alcohol, and endotoxin and accelerates disease progression. That's all I have to say. I've taught you more than I know. I just want to acknowledge the Pancreatic Research Group. Professor Apte is our director and has been for the last 10 years, and she actually sort of leads, she's the leader of the band. She's the organ grinder, I'm the monkey. The other people are sort of either very old, very venerable professors, or the people who do the work, the students and the PhD students. So thank you very much for your attention.